Hello students, today we're going to be talking about chapter 12 or the circulatory system, and this is usually done over a series of 4 to 5 lectures in class. So I'm going to break it down into 4 different tutorials, and you guys can kind of go over maybe 1 or 2 a day so that you're not overloaded with the circulatory system. So these are the different topics we're going to be discussing. There you go on the second slide as well. And we'll start off with an overview of the whole system. So it's made out of, first off, the heart, and that's the pump that is going to be pumping blood into the vascular system, which would be your blood vessels. And the blood vessel is interconnected, so meaning that, if you start at one point, you can pretty much take that whole circulation and come back to that same point. And it's also full of blood, okay, which would be the the fluid that is being pumped by the heart and then circulating within that vascular system. The action or the activities of the cardiovascular system are impacted by the endocrine system. So you have some hormones, for example, adrenaline or epinephrine and thyroxine, for instance, that speed up the heart, for example. It has different effects on the blood vessels. The nervous system also, through the sympathetic and parasympathetic effects, would also have different effects on the cardiovascular system, and the urinary system as well. So the kidneys, obviously, by making more urine or less urine, or for example, by the production of erythropoietin, which is a hormone that stimulates the making of more RBCs. So you can see that there are different ways how all of these different systems can also affect this system that we're talking about. So how do we measure blood, or what are the components of blood? Okay, so blood, if you take a sample of blood and you run it into a centrifuge is basically kind of acts like a dryer, where it's it rotates really fast. And because of that fast rotation, you're able to separate the heavier cells. Those are going to precipitate. So you have about 45% of your blood is made out of erythrocytes, which are your red blood cells, and that is known as the hematocrit value. So the hematocrit value is the percentage of RBCs, in respect or in relative to the whole blood. And then you've got about 55% made out of plasma, and plasma is basically water that is full of everything. So full of plasma proteins, hormones, vitamins, ions, metabolic waste, and a whole bunch of other stuff that we'll talk about. And then in the middle, you have this little buffy coat, which is kind of, whitish, made out of white blood cells and platelets. Okay, so that is the breakdown of what blood is made out of. Okay, so we'll talk first about plasma, which we said it's basically made out of water. Okay, and there's a whole bunch of things that are circulating or that are swimming in that water. For example, electrolytes, nutrients, for instance, glucose, amino acids, and vitamins. We've got waste products like urea, bilirubin, and creatinine. We also have gases like oxygen and carbon dioxide, different hormones and plasma proteins, for example, albumins, globulins, and fibrinogen. Serum, though, which is the same as plasma, but without, so minus the fibrinogen and the plasma proteins that are involved in blood clotting. So if you take serum, okay, which includes all of these different structures, and you utilize that serum to make a blood clot, so you're going to use fibrinogen. You're also going to be using a whole bunch of plasma proteins needed to make a blood clot. When you exclude all of those, you end up having serum. Now here are the different cells of the blood made by this one hematopoietic stem cell. So this is a stem cell. Hematopoietic stem cell means a stem cell that is basically going to make up your blood cells. Okay, but it is uncommitted, so we don't know which blood cell it's going to make. We have 2 kinds, though, of committed stem cells, and those are either your lymphoid stem cell or myeloid stem cell. So the lymphoid stem cells are going to end up making your T and B lymphocytes, while myeloid is going to end up making all of the other cells. So it'll end up making red blood cells or erythrocytes, platelets. It'll also make up your neutrophils, eosinophils, basophils, and monocytes. Okay, so these, unfortunately I cannot really draw a line, but these right here, monocytes, neutrophils, eosinophils, basophils, and these two lymphocytes, those are the different kinds of white blood cells, and we're going to discuss those later on. And other things to kind of keep in mind while looking at this image, again, is, this is your lymphoid stem cell that'll end up making your T and B lymphocytes. This would be the committed stem cell here that is basically going to end up making every single other cell in your bloodstream. That would be the myeloid stem cell. The myeloid stem cell can end up being a red blood cell. In the pathway, it'll make this reticulocyte. So a reticulocyte is the red blood cell precursor, and it's usually only found in the bone marrow. Just a very few amount of it appears in the actual circulation. If too much of it appears in the circulation, that means that the bone marrow is making way too many RBCs at a very fast rate and not giving it enough time to mature from a reticulocyte into an RBC. Platelets, on the other hand, these are not really true cells. Those are just fragments, cytoplasmic fragments of this bigger cell known as a megakaryocyte. And then again, you have all of your white blood cells. So monocytes are the cells, we call them monocytes when they're inside of the blood vessel. When they leave the blood vessel and reach into a tissue, they become known as a tissue macrophage. So monocytes and macrophages are the same cell, just a monocyte is inside of the blood vessel, while macrophage would be in the tissue. The neutrophils, eosinophils, and basophils, those are your granulocytes, meaning that if you look under a microscope, you'll find granules in them. And then you've got T and B lymphocytes, and we'll kind of talk about, more details of these white blood cells as we go through the chapter. Now, first off, our RBCs. Okay, the erythrocytes, that is where your gas is going to be transported. So the oxygen, it will be transported from the lungs to the tissue, while carbon dioxide is transported from the tissue to the lungs. And it has this huge protein. That protein is known as hemoglobin. It kind of looks like a plus sign, okay, where each part or each arm of that plus sign is able to attach to an oxygen molecule. So hemoglobin can carry a maximum of 4 oxygen molecules. Okay, the cell does not have a nucleus. Okay, it does not have any of the organelles. So it lacks mitochondria, DNA, RNA, and that is why these red blood cells cannot, they have a very short lifespan, and they cannot divide to make more RBCs. Okay, the lifespan of RBC is about 120 days, which is basically about 4 months. And these RBCs then are going to die off, and we'll see what happens to them in the next couple of slides. But then, again, because of the lack, it lacks all of these organelles, and that gives it the ability to squeeze itself into these very tiny capillaries in order to perform its function. So here, this is what it looks like. They are made in the bone marrow, in the red bone marrow, and the process of making an RBC is known as erythropoiesis. The process is stimulated by a hormone known as erythropoietin, which is a hormone made by the kidneys. Okay, so the kidney is going to make this hormone. That hormone will circulate through the bloodstream till it reaches the bone marrow, and that's going to stimulate the bone marrow to make more RBCs. Now, what happens, 120 days later when that red blood cell now becomes an old cell and it needs to be, and it's going to die? So the spleen is going to filter and remove all of the old RBCs, and then the liver, and then those damaged RBCs from the spleen will go to the liver, and the liver will metabolize it and break it down. So we'll break down that hemoglobin into heme and globin. So I think there's a picture here. Yep. So actually I borrowed this picture from another course, okay, where we start off here by absorbing iron from the intestine, and that iron is transported to the bone marrow. When the bone marrow is stimulated by erythropoietin to make RBCs, we'll make the RBCs. They'll circulate in the body for 120 days, and then the old red cells are going to be filtered by the spleen and then transported into the liver, and we'll start the whole breakdown of the contents of the red blood cells. So, I know we've mentioned this several times, but the body is a very conservative being, and it likes to recycle things if it can. So the hepatocytes will break down hemoglobin into heme and globin. Globin is the protein part. So globin will break down into amino acids, and then the body can reuse those amino acids for whatever it needs. While the heme part breaks down into iron and biliverdin. So again, iron is something that we can reuse. So you see it being transported back to the bone marrow where it can reuse it to make fresher, healthier RBCs. And then the biliverdin, by the hepatocytes, will change it into bilirubin, and that will become part of the bile that will be transported into the small intestine to help in the digestive process. So going back here, because we want to talk a little bit about iron. Okay, iron, in order to be transported, it needs to kind of be carried by a protein, and that protein is known as transferrin. So ferrin, Fe, that is your symbol for iron, okay, and then trans means to transport it. So transferrin is the plasma protein that transports iron in the bloodstream, and it'll deliver it, like I said, to the red bone marrow, and that can be reused to make new RBCs. Or the iron can actually be stored as ferritin in the liver, or in the spleen, or in the small intestine. And we could kind of store extra iron if we need to, so that when if there is any iron deficiency due to lack of intake of iron in food, we can kind of tap into that iron storage. Now, what are the requirements in order to produce a normal, healthy RBC? Well, we obviously need iron, okay, because it's a component of hemoglobin. And we also need folic acid. Now, folic acid is needed basically for any cell in order for it to replicate. And we also need vitamin B12. Okay, vitamin B12 is needed for the action of folic acid. So the whole process of erythropoiesis, which again is making your RBCs, is really under the control of this hormone erythropoietin made by the kidneys. And if the kidneys ever get the message that there is hypoxia, so low oxygen levels reaching the kidneys have these chemoreceptors, and they're able to pick that up, and that will stimulate the kidney to make this hormone erythropoietin. Erythropoietin circulates to the bone marrow and increases the production of erythrocytes. Okay, that is going to bring up your blood hemoglobin levels up and restore the oxygen levels back to normal. So a little bit on erythropoietin and some clinical issues. So let's say, we agree that erythropoietin is made by the kidneys. Well, what if somebody has renal failure and their kidneys are unable to make that hormone? They are going to develop anemia if they do not take that hormone from an external source. So renal dialysis patients, that kind of lack that erythropoietin, they would have to take it, they have to get it in a synthetic form. An athlete who would abuse erythropoietin, okay, in order to increase the number of red blood cells, which would, the theory behind this is that they are increasing their oxygen capacities and therefore increasing their muscular endurance, which is true. But on the other hand, you're also making too much RBCs, and that's going to make the blood very thick. and they are now more prone or at a higher risk of developing blood clots, stroke, and heart failure. Okay. Testosterone enhances or increases the production of RBCs because it increases the production of erythropoietin from the kidney. And that's why men, generally speaking, have a higher hematocrit value than women do. Now for anemia, know that anemia is really an incorrect term. An means no, emia means blood. So these patients are not bloodless, okay? But it really means that they have a decrease in the oxygen carrying capacity of blood. And that could be due to three different things. Either there's a decrease in the number of RBCs, or the number of RBCs is fine, but the hemoglobin, something's wrong with the hemoglobin, either lower concentration or defective hemoglobin, or it could be a combination of both of these two things. Okay, so for example, sickle cell anemia, that's a genetic disease where there is a mutation that happened in the hemoglobin chain. So as we all know that in order to make proteins, we have the different genes that code for these different proteins. In this genetic mutation, we have substituted just one amino acid in this whole protein chain, and that led to this disease or sickle cell known as sickle cell. And what happens is that during normal oxygen blood levels, nothing really happens to the patient. Patients are symptomless. Symptoms develop if there is stress, for example, or low oxygen levels, and that is going to lead to these hemoglobin molecules that are going to change into what are known as these sickle-shaped cells. And these cells are no longer able to squeeze into the capillaries. They get stuck there, and that leads to severe pain, okay, due to the hypoxia that's happening in the different tissue. Okay, and that destruction that happens to these RBCs can lead to consequently can lead to anemia. So major causes of anemia, the most common cause is iron deficiency anemia, okay, or maybe deficiency of folic acid or B12, which are the three dietary needs in order to make healthy RBCs, or aplastic anemia. Aplastic means no formation, so there is a failure of the bone marrow to make RBCs, and that could be due to maybe radiation or toxicity or cancers that have inhibited the bone marrow. Or it could be that the patient has anemia due to blood loss, okay, due to severe hemorrhage, or it could be hemorrhage over a long period of time. So let's say somebody has maybe colon cancer or diverticulosis, where they are diverticulitis, where they are kind of constantly bleeding for longer periods of time, but there is minimal bleeding. So sometimes it's not even seen visibly in the stools. And that long, that bleeding for a long period of time, that kind of trickle of blood can lead to hemorrhagic anemia. Or like I said, as we mentioned before, if somebody has kidney damage or renal damage due to the lack of erythropoietin, that could lead to anemia as well. And then there's hemolytic anemia, where there is the destruction of RBCs, for example, in the sickle cell disease that we just discussed. Now, moving on to leukocytes or white blood cells, those are important they're part of the immune system, and we divide them according to the absence or presence of granulocytes or granules. So these little granules, if they're visible using a light microscope, we call those granulocytes, and we have three different kinds of granulocytes depending on the color of the granules. So if these granules are purple, okay, or somewhere neutral between blue and pink, we call those neutrophils. If the granules are red, those would be eosinophils. If the granules are blue, those are basophils. Agranulocytes, those are the cells that lack granules in the cytoplasm using a light microscope. And those are either lymphocytes or monocytes. And remember, macrophages are the same as monocytes, but monocytes are inside of the blood vessels. Macrophages would be in the tissue. So what are the basic functions of leukocytes? Each of these cells, I mean, they're all important for for our immunity, but they are kind of specific to different things. So for example, neutrophils are very high during bacterial infections, okay? These are the cells that are your first responders. So if there's anything weird going on in the body, the first cells to arrive would be your neutrophils. They are highly phagocytic cells, and you'll find them elevated, again, as I mentioned, during secondary infections. During bacterial infections. Eosinophils, those are more specific for parasites, so like worms, okay? So if somebody has a white blood cell count and it's elevated and they find eosinophilia, which means increase in the number of eosinophils, that means that that person has a parasitic infection. It could also be increased in the in allergies for as well. Basophils are white blood cells that will be elevated during an inflammation and during infections in general, and basophils have these granules that are full of heparin and histamine. So heparin is a blood thinner, okay, and histamine is a vasodilator. And you want to have those two actions during an inflammation. So if there's an area that's inflamed, you want the blood vessels to dilate in order to bring in more blood to the area, and you want a blood thinner to be available so that the blood doesn't clot, and these blood clots are going to prevent more blood from coming in and help fight off the infection. Monocytes are another example of phagocytic cells, and as I said, when they migrate into the tissue and into the different organs, they are going to mature and grow up and develop into these macrophages, which are very highly phagocytic as well. Macrophages are these huge phagocytes that are able to eat up viruses and bacteria. Lymphocytes, we have two different kinds. We've got T and B lymphocytes. And those are more, they work in both kind of immunities, whether it's whether it is the innate immunity or the more specific immunity. But when you have T and B lymphocytes, especially, that means that, they're more specific. For example, like if all of these fail to work and you need more aggressive attack, you need a more kind of specific attack to these pathogens, the body will start making off these T and B lymphocytes. They can fight off different pathogens, viruses, bacteria, different toxins, and even cancer cells. That would be your T lymphocytes in particular, where, we all develop cancer cells throughout the day, every single one of us, but not all of us end up developing cancer. So these cancer cells are spotted and destroyed by one of the kinds of T lymphocytes that we have. So the phagocytic cells, among all of these cells we talked about, are your M&Ms. So M would stand for monocyte, and neutrophils, and then the other M would be macrophages. Those are the three phagocytic cells among all of these white blood cells. Okay, just to be clear, it doesn't mean that the other white blood cells cannot undergo phagocytosis, because they do, but when we say phagocytic cells or have a high phagocytic activity, it's the M&Ms that we're talking about. Now moving on to platelets. So platelets, again, are not real cells. Okay, they do not have they don't have a nucleus, just like the red blood cells. They're both the same. They're non-nucleated cells. They're actually fragments from this huge cell known as a megakaryocyte. So they're kind of like the cytoplasmic buddings out of that cell. So just pinch off of that big cell, and they circulate in the bloodstream. Now, how does our bone marrow know which cell to make? Okay, so we have these hematopoietic growth factors. For instance, erythropoietin, that is specific for erythrocytes, and we already talked about it, that it is a hormone made by the kidney, It circulates to your red bone marrow and stimulates the process of erythropoiesis. We have other growth factors though, for example, colonies stimulating factors. Okay, they could be either granulocytic or agranulocytic CSFs, and those will stimulate, your granulocytes and maybe your monocytes too. Interleukin, inter means in between, leukin comes from leukocytes. Okay, we have tons of interleukins, so they're kind of numbered. We've got interleukin 1, 2, and 3, and so on. So the example that they're talking about here is interleukin 3, and that's basically going to stimulate the making of various kinds of leukocytes. Thrombopoietin. Thrombo comes from the word thrombus or clot. So you would imagine that a thrombopoietin will end up stimulating the making of platelets. And then the stem cell factor, that is, which is one of the, growth factors, that's going to stimulate the making of different kinds of blood cells, because again, you're kind of stimulating that stem cell at the very top. Now we want to kind of dive into the actual circulation and blood vessels. So we want to differentiate between the systemic and the pulmonary circulation. In order to understand that, we want to take a look and see what are the different parts of this image that we're looking at. That right here is your heart. This represents the capillaries in the lungs, so that would be your pulmonary circulation, while this represents the capillaries in the systemic circulation or in the rest of the body, so your upper limb, trunk, abdomen, pelvis, lower limbs, and basically everything besides your lungs. So what happens is that the deoxygenated blood, meaning that the blood that does not have oxygen in it, or oxygen poor blood, will be transported from the right ventricle, pumped from the right ventricle into the pulmonary artery. And the pulmonary artery will take that deoxygenated blood to the lungs, where it will pick up oxygen and then come back through these pulmonary veins, and I'll pour what is now the oxygen-rich blood, or we also known as the oxygenated blood, into the left atrium. That would be your pulmonary circulation. Okay, so you start off here in the right ventricle, and then you do that loop and you end up in the left atrium. Okay, the systemic circulation would start here in the left ventricle, where you pump that oxygenated blood into the rest of the body, and all of the cells will take up the oxygen and utilize it for whatever function it's doing. And then now that deoxygenated blood will go back to the heart, and all of that will pour into the right atrium. This represents your systemic circulation. Okay, so pulmonary circulation, you start off with deoxygenated blood, and then you end up having oxygenated. While in the systemic circulation, it's the exact opposite, where you start off with oxygenated blood and you end up with deoxygenated blood. And all of those circulations obviously are made out of blood vessels. So we want to get a better understanding of the blood vessels. We've got arteries, arterioles, capillaries, venules, and veins. So arteries, those are the big blood vessels. And we, to understand what the difference between an artery and a vein, you really want to understand the direction of blood flow. So an artery would be taking blood away from the heart. Okay, so an artery with an A means you're taking blood away from the heart, regardless of what that blood is. So whether it is oxygenated or deoxygenated, that doesn't really matter. Just look at the direction of blood flow. So you'll see here, this right here is taking blood away from the heart, and that is why we call this a pulmonary artery. While this right here is taking blood into or towards the heart, and that is why we call these pulmonary veins. Although we're kind of used to having, the veins in blue and the arteries in red, which is the general rule, but there are a couple of exceptions. One of those exceptions are your pulmonary blood vessels. And that's why when you name blood vessels, you name them according to the direction of flow. So artery would be away from the heart. Veins and venules would be towards the heart. Okay, so your arteries again are your big blood vessels, and then it'll start to divide into smaller arterioles. Those arterioles end up taking blood to these capillaries, and capillaries are blood vessels that are made out of one layer of simple squamous epithelium, just like the air sacs are, because you need that one layer of flattened cells to help with the exchange of gas. And now all of that blood will be delivered back to the heart. We'll start gathering them through these smaller venules, and then end up making bigger veins. And all of that deoxygenated blood again will be pumped or dumped into the right atrium. Okay, so in general, yes, arteries do carry oxygenated and veins carry deoxygenated blood, but there is definitely an exception to that, like the pulmonary artery. So again, you want to take a look at the direction of flow as opposed to the color of these blood vessels on a model. Okay, in our bloodstream, in our bodies, our veins and arteries are not color-coded. This is a distribution of the systemic blood flow, not in numbers. You don't have to worry about these numbers, but I just thought it was really interesting that at rest, okay, we have about 5 to 6 liters of blood. Okay, and you'll find that the most of that blood is actually in the abdominal organs. So about 1/4 of it is in the abdominal organs. You could see, for example, about 1/5 of it is in your skeletal muscles. The brain has only 13%, and so on. This distribution of blood flow, though, can definitely change. So remember, this is at rest. But if you're exercising, the blood vessels are going to shunt the blood a little bit differently. So it'll start, the blood vessels going to the abdomens are going to constrict, okay, and shunting that blood maybe to your skeletal muscles, to the heart, and to the brain. So you'll find that the blood vessels in these 2 organs, brain, heart, and skeletal muscles, will dilate in order to bring more blood to those areas. And that is how we can change blood flow. Okay. And that is what we're going to be talking about next. So your blood flow and the relationship to our blood pressure and resistance to flow. Okay, so flow is how the volume of blood moved per unit time, and we measure it in milliliter per minute. Okay, and it is affected by pressure. So the pressure of blood, which is basically due to the hydrostatic pressure. Hydrostatic pressure literally means the pressure that is made due to water flow. Okay, so since plasma, which is part of our blood, is made 90% of water, so the pressure, that blood pressure, is due to the hydrostatic pressure, due to the flow of water in our blood vessels. And that is because of the pumping of our ventricles. So as the ventricle pumps the blood, the stronger the contraction, the stronger or the higher the pressure, that would lead to a faster blood flow. Okay, if you look at the resistance, though, that's a little bit different. Okay, so when you put more resistance against your blood flow, that is obviously going to slow it down. And resistance is due to the friction against your blood vessels. So as blood vessels constrict, that is going to lead to higher resistance. That means that the flow is going to decrease. Okay, so blood flow is related to both of these factors. So you have F stands for your blood flow, where it is directly proportionate to the pressure, change in pressure, while it is inversely proportionate to resistance, meaning that if you increase pressure, you increase blood flow, but if you increase resistance, the blood flow will decrease. Now, resistance, which is one of the an important factor determining blood flow to an organ, resistance on its own is determined by 3 different things. Okay, the most important would be the radius of the tube, the inside radius of the tube. So how big or small is the radius of that blood vessel? Again, a blood vessel that is dilated, okay, means a bigger tube that is going to decrease the resistance but increase your blood flow. While if you constrict your blood vessel, that is going to increase resistance and decrease the blood flow. Okay, so that is your most important. Other factors, though, that do relate to resistance would be the thickness or the viscosity of blood. So the thicker the blood, the more resistance or the more friction between the blood and the blood vessel, and that will lead to higher resistance and lower blood flows. For example, let's say somebody who's dehydrated, okay, and now they have thicker blood, and it's kind of hard to kind of push that thicker blood into the different organs. Or the number of RBCs. Okay, somebody that is anemic, for example. Okay, they have lower numbers of RBCs. That is going to make their blood much thinner. That decrease in resistance is going to increase their blood flow. Last of these three determinants would be the length of the blood vessel. So a longer blood vessel will give you more resistance, and that would decrease the blood flow. This right here is a section in the heart, and we are going to go through the whole blood flow. This is your superior vena cava and the inferior vena cava. Those are going to dump the deoxygenated blood into the right atrium, then through the tricuspid valve into the right ventricle. That will pump the blood through the pulmonary valve into the pulmonary artery or pulmonary trunk that divides into the two pulmonary arteries. It'll get oxygen from the lungs and then come back to the left atrium through 4 pulmonary veins. So these are 2 on this side, and you have 2 on the other side. Then from the left atrium through the bicuspid or the mitral valve into the left ventricle. Then the left ventricle pumps it through the aortic valve into the aorta and then to the rest of the body. We're also going to look at the different heart layers. So there's the epicardium, which is the outer layer, which is the outer layer on the outside. Okay, and we have, the parietal peritoneum. There's the parietal pericardium. There's the visceral pericardium, and in between there is the pericardial sac that is full of the very, very thin amount of pericardial fluid. And then you have the myocardium, and that is that's the actual cardiac muscle fibers. And then the inside of the blood vessel, the inside of the, all these chambers are lined on the inside with a smooth endocardium. That endocardium is continuous with all of these blood vessels. So these blood vessels are lined by the same lining as the inside of the chambers. So the endocardium, as I said, that the endocardium would be the inner lining of the heart, and when it gets into the blood vessels, we'll call them endothelial lining of the blood vessels. The cardiac muscle fibers or myocardium. These cardiac muscle fibers are branched out fibers. You'll find that the walls are connected, or the cell membranes are connected by these intercalated discs with gap junctions. So in order to facilitate the transmission of electricity, in order for the contraction to happen at once. Okay, our heart pumps about 3 billion times in an average lifespan without resting. Okay, and the heart cells, or the myocytes, the myocardial cells, have a very limited ability to replace themselves. Okay, so only about 1% of our muscle fibers are kind of replaced per year. Now for the blood supply. So the heart, just like any other organ, it also needs oxygen, and it needs to get rid of its carbon dioxide that it makes. And the heart does not absorb oxygen from the chambers. It has its own blood vessels, and those are known as the coronary arteries. Now, actually, the very first branch out of the aorta would be coronary blood vessels. So the heart has to take care of itself before it gives blood to the rest of the body. So you have the right and the left coronaries, and those will branch out and provide or supply the whole heart with its oxygen, and then the deoxygenated blood will gather back into these veins, and all of them will dump the deoxygenated blood into one big pool of deoxygenated blood known as the coronary sinus. And the coronary sinus enters or empties that blood into the right atrium. So the right atrium really gets blood from the superior vena cava, inferior vena cava, and the coronary sinus. Now for the innervation of the heart, it has sympathetic and parasympathetic fibers. Okay, the sympathetic fibers that innervate the heart release norepinephrine, and you'll find that the heart cells have beta adrenergic receptors in order to respond to that epinephrine, while the parasympathetic fibers are going to release acetylcholine, and the myocardial cells have cholinergic receptors of the muscarinic type. We'll see here the sympathetic cells or the sympathetic nervous system on this end, while the parasympathetic nervous system on this end. And this kind of represents the heart, atria on top and the ventricles on the bottom. So the sympathetic, you see here that we've got beta adrenergic receptors both on the atria and in the ventricles. Okay, they would respond to epinephrine coming from the norepinephrine coming from the sympathetic nerve fibers, and the epinephrine that comes from the adrenal medulla through the bloodstream. Okay, so these beta adrenergic fibers respond to both of them. And again, these beta adrenergic fibers are found throughout the whole heart, atria, ventricles, and in the middle, in the AV junction. While the parasympathetic fibers, we only have these muscarinic receptors in the atria. We do not have any receptors to acetylcholine in the ventricles. So the parasympathetic can only inhibit the atria. While on the sympathetic side, we're able to stimulate the atria, able to stimulate the junction, so the transmission of the electricity from the atria into the ventricles, and we could also stimulate the ventricles. Okay. This ends the first part of the cardiovascular system, and I'm going to, I'll be recording different parts and uploading them for you guys kind of on a daily basis.